Okay, so in section 1, we're going to be looking at identifying DNA as the genetic material. So when we end up looking at this, one of the first people that really did anything was a scientist by the name of Griffith. And what Griffith ended up doing is he did an experiment. He wasn't trying to figure out what carried genetic material. He was trying to figure out what type of pneumonia was the most deadly. And so he had 2 strains of pneumonia that he was wanting to test. And what he ended up finding was that he had, with those two strains of pneumonia, he had the one that was the S strain was smooth, R stood for rough. And what he ended up doing is he was injecting the mice with these 2 strains of bacteria, and he ended up finding out that the bacteria that was the S strain, when he would inject it in, it would kill the mice immediately. And then the R strain, it didn't kill the mice. And so he decided, I'm going to heat the S strain. And as he heated that S strain, what he ended up doing is he ended up seeing that it didn't kill the mice. And so then he hit upon the idea of, I'm going to take the dead S strain and mix it with the live R strain. Neither one of them would kill the mice. And so he ended up injecting that mixture into a mouse, and the mouse ended up dying of pneumonia. And so when he did a little mouse autopsy and looked to see what type of bacteria it was, there was live S bacteria in the mouse. And so that is what he ended up seeing and was able to find. So that is what he referred to as transformation. And he saw the material was passing from the deadly S strain to the live R strain. And so he said something had to be doing that. And what he ended up saying was that thing was what carried the genetic information. So that led to other scientists that were then trying to determine what actually carried the genetic information then. And so that led us to Avery and his team. Avery did the same experiment as what Griffith did. He was trying to see would transformation happen. And so he did a test where he ended up testing multiple things that people thought carried the genetic information. And he would break those things down. And if he ended up not seeing transformation occur, he knew that the thing that he broke down was what carried genetic information. So long story short, the thing that finally was broken down and did not allow transformation to occur was DNA. So when that happened, some of the science community was like, yay, we know what carries the genetic information. Other parts of the science community ended up saying, yeah, we still don't buy that it is that DNA is what carried genetic information. So then that leads us to Hershey and Chase. Hershey and Chase decided that they were going to prove once and for all, here's what carried genetic information. So they did an experiment where they made a virus radioactive, and they knew that viruses infect by putting their genetic material into the cell. Well, a virus is made up of it's made of protein and DNA, and so that is what they ended up doing, is they ended up making the protein and DNA radioactive. And so then they were able to see what was actually put into the bacteria. And when they ended up finding the bacteria at the end, they ended up seeing that it was the radioactive signature for the DNA that was in there, and so they ended up saying that DNA is what carried genetic information once and for all.
So the jobs of DNA, there are 3 specific jobs that DNA has within organisms. The first one is they're going to store genetic information. That should be a pretty obvious one. Another one is it's going to be able to copy the genetic information, and then the third is it's going to transmit genetic information. So I'm going to share with you guys what those things mean and give you some examples of those. So within a cell, any time the cell needs to make any product, it's going to rely on the DNA to be able to provide the instructions on how to make that product. So when I say product, I'm talking about like proteins and things like that we'll talk about later in this chapter. So when it needs to make a protein, that the instructions on how to make it, they're located in the DNA. So an example of this in real life, that would be how the DNA is a place to store information, would be like you could think of it as your textbook has information, tons of information on biology in it that it's just waiting there for you to be able to use and find that information for that will be helpful for the class. The next one, how does DNA copy information? So anytime the cell gets ready to divide, the DNA needs to be copied so that the 2 new cells can have the same information. So once again, use that example of a textbook, each and every one of you have either a digital copy of the textbook or an actual physical copy of the textbook, and I don't have to worry about, do you guys have the same information in this book compared to this book? I know all the information is going to be the same, so it was copied and passed on to you guys. And then the final example of how does it transmit information. So what does that mean? It means that anytime that the cell makes a copy or you reproduce, you are transmitting, you're passing on that genetic information to that next cell or the next generation. So you're passing it on and being able to use that. So once again, for this example, if we are using the textbook example, I've passed those textbooks on to you guys, and I know that the information is in there, and you guys are going to be able to use that to help you function within this class. So that's the end of the notes for section 1.